Today's video I'm going to be demonstrating how to perform an independent groups t-test or a t-test for independent samples. And I've got data that are simulated from an actual study that was published in 2004 by Brody et al. And what Brody et al. Examined was brain sizes between smokers and non-smokers. And in particular, in this example, it's going to be based on the frontal lobe sizes of these participants in the study. So how you place the data into SPSS is pretty simple. You have one column that includes your dependent variable data, which are measured on an interval ratio scale. And it goes from 7.30 in this data set all the way down to 3.70. So there's a total of 36 participants in the sample. And then we've got our grouping variable next to the frontal brain volume variable. So grouping, I've got two groups, zeros and ones. Got the ones at the bottom, zeros at the top. And zeros are people who are non-smokers, and ones are smokers. And how I've labeled that, you can see in variable view by clicking that tab at the bottom. And I've got values here, so 0 non-smokers and 1 smokers. And I did that by adding values here and then adding through this option here. Okay, so the analysis itself is quite simple once you've got your data entered. So dependent variable here, independent group here. It wouldn't matter. You could have your grouping variable here or your dependent variable here. It wouldn't make any difference. So go into Analyze and Compare Means, Independent Samples t-test. So we've got frontal brain volume here. That's our dependent variable. Put that into Test Variables window. And then we've got group, and put that into the grouping variable box. And then SPSS is going to ask you to define the groups, which is simple enough. Click on Define Groups, and then press 0 for group 1 and 1 for group 2. So we've got our non-smokers and our smokers. Click Continue. And I'll note that it wouldn't actually matter whether you have zeros and ones here, whether you used 1 and 2 or 3 and 4 or 10 and 20. Any number would actually be useful in this grouping variable. 0 and 1 is probably what most people use in this case. So click OK. And the output is produced by SPSS. I'll just move this to make the output more visible. Alright, so the first box that is visible in SPSS is the group statistics. And we've got frontal brain volume, group non-smokers and smokers. So sample size in non-smokers was 17 and in smokers 19. And then we've got the means for frontal brain volume. It's not exactly frontal brain volume that Brody et al. Measured. They measured a ratio of frontal lobe volume divided by global brain volume, but we'll just say brain volume for the purposes of this example. So non-smokers had a mean frontal lobe volume of 5.3 versus smokers with a frontal brain volume of 4.3. All right, and the standard deviations are in the column next to the means, and we've got a standard deviation of 1.06 for the smokers and 0.946 for the smokers. So we can tell by looking at the means numerically, there's a difference equal to about 1. 5.3 versus 4.3 rounded. And the standard deviation is about 1. If you average that, the standard deviation is about 1. So the next table that SPSS produces is the actual results of the t-test, but the first thing that it produces in this table is Levene's test of equality of variances. And this is testing the homogeneity of variance assumption. So t-test assumes, the independent group's t-test assumes that the variance, or expressed as a standard deviation are the same in both samples. Now, they don't have to be exactly the same, but they have to be the same to the extent that they're not statistically significantly different from each other, even though t-test for independent groups is relatively robust to violations of this assumption. You should still test it and report it in a result, in a report. So what Levine's test of equality of variance equaled in this analysis is an F equal 0.174. It's a very small F value. And the significance of this F is 0.679, which is not statistically significant. If you get a p-value here, a significance p-value of 0.05 or less, you would say that the variances or standard deviations are not the same in all probability. They were probably sampled from different populations, and so we become concerned about interpreting our t-value and our p-value if the homogeneity of variance assumption does not hold. But in these data, because the significance value is not 0.05 or less, in this homogeneity of variance Levine's test of equality of variance test, we're confident that we can interpret the regular t-value and the significance level associated with this t-value. So t is equal to 3.092 with a degrees of freedom of 34, and the significance level is 0.004. So because this value is less than 0.05, we would reject the null hypothesis of no difference between the means and state that in all probability that the difference in brain volumes between frontal brain volume between smokers and non-smokers is statistically significant, p equal 0.004. So only in about 4 out of 1,000 samples would we expect to get this kind of result as big as this, and it only occur by chance. So in all probability, this result is not a chance finding. I'll note that underneath the equal variance assumed, SPSS produces also an equal variance is not assumed result, which it produces a t-value of 3.072 and adjusted degrees of freedom of 32, and the significance level is the same thing. So basically, the results are the same whether you look at the equal variances or not equal variances, and that's because the variances are basically the same. The standard deviation, in this case, the variances aren't outputted. You just have to square these values to get the variances. But these standard deviations are very similar. And that's why the two t-values are the same, or very close to the same, and the significance level. SPSS also outputs the mean difference between the two means. So mean 5.32 versus 4.29 gives us a mean difference of 1.3467. The standard error associated with that mean difference is also outputted by SPSS, 0.334. And then it gives confidence intervals for of the difference between the means. It's quite rare to see that reported in a report. People typically report the actual means, the standard deviations, the t-value, the degrees of freedom, and the p-value, and then they tend not to report any of this extra information. But what SPSS very unfortunately doesn't output is the last key piece of information in a analysis based on mean differences in two independent groups, and that's called something called Cohen's d. And SPSS very unfortunately does not output this. It's a real disappointment. It would take very little extra computational power for the analysis to be done, but SPSS is just hasn't done it. And Cohen's d has been around for years. Basically, Cohen's d is a measure of effect size. I'm just going to speak about it very briefly in this video, but the difference between the means, which we know to be about 1.03, which is here, and it divides it by the, you divide that value by the average of the standard deviations. And I can tell you that the Cohen's d in this case, which you have to calculate by hand, ends up equaling something like 1.03 or 1.06. So a Cohen's d that's relatively large by standards that are used to evaluate Cohen's d. Anyway, that's how you perform an independent samples t-test in SPSS. Bye.